Hi everyone, welcome back to my channel. My name is Jeffrey. Today we're going to go over cell death. So there are two main processes of cell death. One is necrosis and the other is apoptosis. Now you've probably heard of both. When you hear of like the flesh-eating bacteria type deal, it's kind of like leading to necrosis of your tissue. And apoptosis, you've probably heard this like 15 billion times if you've ever taken any biology course ever. So let's just start with necrosis. It's characterized by cell swelling, by your plasma membrane damage, and your DNA degradation. So this process is a little bit more messy than apoptosis. As you can see, your cell swells. Eventually, it's going to if you have a normal cell, it's going to increase in size, and then eventually it's going to pop, right? And when it pops, everything inside of that cell comes rushing out, including things like lysosomes and proteasomes. And that actually hurts the surrounding environment because things like lysosomes have very, very low pHs. So that's why you would have, from plasma membrane damage, you would get some really bad effects to your neighboring cells. Okay? I hope that makes sense. All right? And then, so when we're looking at apoptosis, it's a little bit less messy than necrosis. Basically, there are some more steps to try to ensure that this type of process is more localized and dealt with in a more efficient manner without destroying the cells around the cell that you're trying to kill. Okay, so it's characterized by cell shrinkage. I underlined these in red. So these are things that you have to know for necrosis, but for apoptosis, the must-knows are cell shrinkage, so as opposed to cell swelling. That's how you can characterize the difference between the two. You have plasma membrane blebbing. Blebbing is what you probably expect as you hear the word. Normal plasma membrane looks like this. Maybe blebbing has some increased blebs around. You have chromatin aggregation, nucleus fragmentation, and oligonucleosomal DNA fragmentation. So basically what's happening right here is you're degrading your DNA in a very stepwise fashion as opposed to random DNA degradation all over the place. So what you're trying to do is you're trying to make sure that this DNA is destroyed once and for all and that no other things can use that DNA for replication. Because for apoptosis, something is wrong with the cell and you want to kill it for good because of its DNA, of the proteins that it's making. So it makes sense that you want to destroy this DNA in a very stepwise, in a very orderly fashion. And then you also have this thing called the caspase cascade activation. This is probably one of the key characteristics that separates apoptosis from necrosis. Now, let's just talk about the two different pathways for apoptosis. Now, I'm going to walk with you guys step by step through this pathway because these intrinsic and extrinsic pathways are extremely, extremely complex. So let's just take a look, all right? So the first thing that you have is an apoptotic stimulus. And what that does is that death signal, which is from DNA damage or anything else, that leads to this apoptotic stimulus being formed. And then what happens is that you're going to have a pro-apoptotic protein regulated, such as BAX. So this apoptotic stimulus could be BAX, and that goes to your mitochondria. It's going to release your cytochrome C from inside of your mitochondria. If you guys remember from MCAT studying, for example, cytochrome C outside of the mitochondria is a clear sign for apoptosis. So when the cytochrome C gets released, it's going to stimulate this activation factor called APAF1. So the two are going to bind together, and that is going to stimulate the assembly of a thing called a apoptotisome. Apoptosome? I don't even know how to pronounce it, but it's going to make this complex. So as you can see, it's a bunch of just APAF1 factors with cytochrome C, and they're just arranged in this circular fashion. And that's going to recruit recruit and activate your procaspase 9. And procaspase is just another type of those cascades, and that's going to lead to your caspase cascade, and that's going to lead to all of these factors going on in here, and then eventually you're going to undergo apoptosis. So now for your extrinsic pathway, this is a little bit different. So extrinsic pathway, what happens is it's actually extracellular. So you have something that's coming out from outside of the cell, and it's coming and binding to your cellular receptors onto your cell. So in this case, you're going to bind this ligand, this Fas ligand, onto the Fas receptors. That is going to recruit your death domain receptors or adapters. So in this case, it's called the FADD, and that leads to the formation of a DISC complex. That's a death-induced signaling complex. And as that DISC complex is formed, that's what's going to trigger your caspase cascade in your extrinsic pathway. So it activates the initiators for your cascades, and then that leads to your caspase cascade, which leads to your apoptosis. Now, I've said cascade and I've said cascade a lot of times, so you're probably wondering, what the hell is a caspase? The role of a caspase, well, let's just first talk about what a caspase is. So they're first synthesized in an inactive form, in a procaspase form. So if we look in the intrinsic pathway, you can see that it's in an inactive form here, because you don't want to induce apoptosis just randomly. That won't be good for any of your cells. So it's synthesized in an inactive form, and then it's cleaved to activate. So in this case, for the intrinsic pathway, it's cleaved by the apoptosome, and then that activates the cascade, the caspase, and then that leads to the cascade. Hopefully that makes sense. That has a similar function over here in the extrinsic pathway. So you can see that procaspase 8 or 10 is going to be cleaved, and then that's going to lead to your initiation of your apoptosis. And now I'm going to touch on where it targets. You can see it targets a lot of different things in your cell. So it targets your cytoplasm, your nuclear proteins, and your caspase active DNAs. So let's just break down everything in here, right? So you want to target the cytoplasm because, you want to break down the organelles in the cytoplasm. You want to target your nuclear proteins because you want to break down everything that's synthesizing DNA, because that cell is damaged or that cell needs to die. Your caspase active DNAs, what this means is that this breaks down the DNA for your caspase activation. You essentially want to just kill everything in the cell, right? So that's why you have a caspase active DNase. You also cleave your cytosolic protein, so it's the same thing as targeting your cytoplasm, and it destroys your nuclear lamin. Your nuclear lamin is important because in your nucleus of your cell, your nuclear lamin is actually what keeps that nucleus intact. So when you destroy the nuclear lamin, you're going to be destroying, or you're going to be opening up that DNA to aggregate, to undergo fragmentation, because your DNA in your nucleus is most stable inside of the nucleus. If you break the nucleus, then your DNA is going to become unstable and it's going to fall apart, and then you have the other things that break it down as well. Okay, so now you're thinking apoptosis is great. What are some of the physiological processes? Like why is this important? And it's important in embryological development. What that means is if you're making your digits of your fingers, for example, that's going to actually make your fingers. So in your embryo, your hand kind of looks like this, kind of like a mitten. And what apoptosis does is that it prevents the webbing of that finger. So then now you get a hand or whatever. So stuff like that. So you're not having that webbing in between your fingers. You also have normal tissue homeostasis. So if you guys ever heard of anything related to skincare, it's like, oh, your epithelial tissue, the skin of your face has a lot of dead skin on it, so that's why you need to exfoliate, things like that. You just want to, for some tissues, they need to undergo apoptosis after a certain time period because they replicate so fast. And because they replicate so fast, you want to kill some of those cells because they might become cancerous. They might have some mutations, some point mutations, because of how fast they're replicating. And then problems if apoptosis doesn't work properly, you can have some autoimmune disease or malignant neoplasia that results in the accumulation of unwanted cells. So it essentially goes back to what I was saying before. There are some cells that you do want to kill after a certain period of time, for example, epithelial cells. So these are some problems if your apoptotic pathway doesn't work properly. And now you're like, okay, great, I understand apoptosis, but how do you even start apoptosis? Like, how do you stimulate it? Or how does that, like, how does the cell know to undergo apoptosis? So that's your question, and the answer is going to be balance. You're going to have two different things. You're going to have your pro-apoptotic proteins and your anti-apoptotic proteins. And your pro-apoptotic ones are your BID, BAX, and BAC. These lead to the degradation of the mitochondrial membrane. So if we look back up here, remember that BAX in the example that I gave was your apoptotic stimulus, and it breaks down your mitochondrial membrane, right? And then your BCL2 is going to be your anti-apoptotic proteins. An example would be the very famous p53 tumor suppressor. So what it does is you're essentially just trying to create a balance between these two different types of classes of proteins. And if you have your BID-BAX having a higher concentration inside of the cell than your BCL2, then you're going to have apoptosis. And if you have a higher concentration of p53 or your BCL2 proteins, then you're going to survive. So I hope this explains the process of cell death, the difference between necrosis, apoptosis, and inside of apoptosis, the intrinsic and extrinsic pathways, and how apoptosis happens. All right. If you guys have any questions, leave them in the comment section below. I'll get to them. And